Morgan Heller several years ago, which we're happy to kind of restart after some pause because of the situation. And it's really an honor to have Professor Sharpee here, representing what we strive to do here in ELSC as an interdisciplinary center. Her work spans from theory through experimental work, and even to things that are more philosophical about the way we represent the world and how we create invariances about the world. She started, like some of you, in physics with a master's in physics, and then went on in Ukraine, and then went on to Michigan to do a PhD in theoretical physics, and since 2001 has been in California at the Salk Institute in the University of California in San Diego. Professor Sharpee, needed to say, published many very influential papers in the field of theoretical computational neuroscience. She's a fellow of the American Physical Society, won many awards, recently the Leno Award in Computational Biosciences. She mentored many students who went on to either have a position in other universities or in industry, which is also something that we pay attention to here. So with that, I'll just clear the stage for you to speak about reading out information from large neural population without loss.
And the invitation. So today it will be more detailed theoretical talk, but I hope still important, and I'm looking forward to the discussion that was very productive on Tuesday, and I hope that there will be also a lot of questions, a similar number of questions today. So I will describe a result that I think is simple, but one of the claims is that you can, how to read out information that is available in neural population. The claim is without loss, under assumptions about, some assumptions about how neural responses are structured. So today I have prepared a three-part talk. One is the equation, a rather simple equation for how to read out neural responses without loss, and from large neural populations. And then there are two aspects of two challenges. One is large neural populations, which I think addressed in number one. And the second challenge is that the stimuli are three-dimensional. And we do not have a full solution, but what the theoretical results suggest is that you can decompose it into parts. And this part is can be solved or points to the importance of modulation. We will see how that comes out from the theoretical equation. And the last part will be practical. So it turns out that if we have to to read out information from neural population, how to deal with high-dimensional stimuli, we need modulation. And if we are working in the presence of modulatory signals, then it turns out that the theory makes a very specific prediction for the role of inhibitory neurons and how this modulation has to be injected into a local circuit. So that's the plan for today. So starting with the readout. So it's a classical problem that has a long history, and I'm sure many of you use this approach called population vectors. A very simple and influential idea goes back to Georgopoulos' work, and he did it in the motor system, so I will use that example. But it's used everywhere in many different systems. So in this case, it's a monkey experiment, and the monkey is making movements in different directions. So the monkey can make a movement down to the left or up and right, and whenever the monkey is making different movements, they are recording from a single neuron at a time. So right now, what is shown here are responses of a single neuron as the animal is making diverse movements. And one can see that for that neuron, whenever the animal is making movement in this direction, the response is strongest. So one can characterize a diverse set of movements by specifying that for that neuron there is a preferred kind of movement direction or stimulus direction. We will call it W. And so by observing the animal in multiple conditions, you assign to a neuron one vector that specifies its selectivity. So that's the encoding part. We observe for a long time, and for each neuron we describe what is its role in a movement. Now the decoding part uses the same construction in reverse. You make an instantaneous observation, but on the large or multiple neurons. So in this case, how to read out what the movement was, we say each neuron stands for a certain direction of movement, and then we average by how many spikes that neuron produced to get a population vector. It's a very simple construction, and I would say the miracle in this paper was that what they showed is that using the simple construction, one can read out to a high accuracy about what the animal movement will be based on neural activity. Any questions about this setup? So now this is very popular. You see, you can use it to decode place cells in the hippocampus, motor neuron activity here, in eye movements, in visual neurons. In this case, this vector represents a receptive field or a stimulus pattern that the neuron is responding. So, but this population vector has an assumption, and the key assumption is that neurons tuned to the same stimuli can be averaged. So remember, that's the assumption. So if the neurons have the same W, then I can just add their spikes, and that's all that matters. And now this assumption was tested and found to be false. So there is I'm quoting one paper, but then there are other papers that similarly have came to a similar conclusion. I happened to be in the same office with Leslie Osborne and Stephanie Palmer when she was working with Steve Lisberg and Bill Bialek on this problem, and they asked if it is true that you can average neural responses that are tuned to the same, say, stimulus, then let's check this in detail. So they constructed, through some synthetic manipulations, a set of a population of neurons that are tuned to the same direction. They have different tuning widths, and then they observe how would this population respond to different stimuli. And they're comparing whether it matters. So technically, according to the population vector construction, you can just sum up the responses of this population, and how strongly this population responds, that's how it will represent the stimulus. So that's the curve that will be here, and this is how much, and they quantified how much information this population convey if you just sum the neural population activity, and the x-axis is the number of neurons. So one can see that there are two curves here, and I will explain what this curve is, but this other curve is how much information does this neural population convey about the stimulus if we keep track of which cell fired on a particular trial. And they call it as words. So one can see that if you keep track of which neuron fired when, then there will be a substantial information loss between what the neural population can convey and our simplified procedure based on the population vector or population count. Any questions? No? So this reminds me of the theoretical neuroscientist dialogue. So this is from a sci-fi movie. They're landing on an alien planet, and a theorist says you can't do that. Just open the hatch, and the experimentalist breathes air and says, seems okay. So this is our setup, where population vectors, experimentally, we do it, it works. Accounts for movement activity, it works. And then the theorists come over and say, no, you can't do that. You're going to lose a lot of information. And they say, well, we don't care. It works. So my observation is to resolve these two, to say that in this argument, that actually both sides are correct. And the solution will be that population vector is actually quite accurate, but it needs to be modified. There will be one more factor here that can resolve, can address why the theorists say this is not a complete solution, and at the same time, why experimentalists say that most of the time it's okay. So that's
question.
Yeah.
So the issue of counts versus words is here, but even with counts, this may not be the optimal way to decode.
Well, okay, so the count there are many ways to be suboptimal.
So when you show the count,
yes,
you actually mean decoding with this method, the count.
yes.
But there may be more optimal ways to decode the movement from only the count.
So you're saying, okay, so there is a specific way, so this is full information. We don't argue
So
about
this
that.
information of the count is not relying on this population vector.
It does. No?
So this is optimal decoding from information?
Well, I'm going to say it's
No,
not
no,
optimal.
if we just have count.
If you just have counts, you're saying there is a better way to decode. And I will derive and I will show when this is optimal based on counts and when it's not optimal. And the purely counts, we have not found an optimal way, but with the modification, then you can do count. So the intuition is, so let's see how, anyway, so how to, so the claim is that you can, maybe this addresses, maybe my slides are a little bit out of order, but so let me derive the equation. So canonically, or usually when people talk about decoding, we talk about tuning curves. And this is one way to represent a population, responses of a neuron, meaning this is its preferred stimulus, and this is how it deviates from the preferred stimulus, and this is how the spike probability decreases when you deviate from the preferred stimulus. This is one approach. An equivalent approach, you can go back and forth between these two, is to use the so-called linear nonlinear model. And it says, I have a preferred stimulus pattern, shown here, and this is my spike probability as a function of how similar that stimulus is to the receptive field. And we have a nonlinearity. So if I change the stimulus, this will change the shift. If I change the optimal stimulus pattern, that changes the orientation. And if I change parameters of this nonlinearity, it will change the tuning curve. So there's a one-to-one correspondence between these two approaches. And in the simple model, the nonlinearity here has two parameters, threshold and the slope. If you change the slope to be more shallow, then the tuning curve will change. So there will be a smaller change between the maximum and the background. And if you change the threshold by keeping the same slope, then maybe the maximum curve will go down and the floor will also go down. But there is a one-to-one relationship between parameters of this nonlinearity and a family of these tuning curves. So it turns out that if we use not the tuning curve description, but this equivalent description in terms of linear-nonlinear model, and so we describe the neural response or the probability of a spike given a stimulus as a logistic function. And here, under the argument, we have a dot product between a preferred pattern W times the stimulus S, and a beta parameter controls the steepness of this curve. So if I write down this neural response in this way, then the claim is that there is a sufficient statistic for the neural populations. Right now we will start with neurons being conditionally independent given the stimulus, but you can rewrite it in a form, this population response, that has a function of the stimulus, function of the neural response, and then a product between stimulus and response. And the function that couples this product is the population vector. The only thing is, it is known as a sufficient statistic. It is a population vector, but it has an extra factor. And the extra factor is the steepness of this nonlinearity. So it says, if you think of the population vector as a democratic solution, meaning each neuron gets its vector, and then they vote for the with its number of spikes for what is the stimulus, how close they're representing. So, yes, no. So now we have a weighted democracy, weighted by expertise, meaning neurons that have larger beta, meaning they are more reliable, they have a sharper nonlinearity, will get a stronger weight.
Question. So why can't I absorb beta in the definition of W?
You can. So the way, at least in the Georgopoulos original paper, the W was normalized. But the point is that the W shouldn't be normalized, and the length of this receptive field represents the reliability of the stimulus. So what we are saying is, let's see here, that using this procedure, you can actually reproduce the dichotomy that was in this paper that I quoted. So, for example, we can create a synthetic population of neurons. They are all tuned to the same stimulus. They have a diversity of tuning curves around the stimulus. And if you use the classic population vector, which in this case is the count, the information will be lost compared to the full information calculation. But you can capture that loss by adding just one parameter, which is the how noisy is the nonlinearity in the neural response. And curiously, this threshold does not come into the decoding equation. So one could ask, how can this be? And basically, in a simple case with two neurons, we can have 4 different patterns. So the claim is that it's a strings. So with 2 neurons, we have 4 patterns: 0, 0, 1, 0, 1, 1, and 0, 1. But imagine that the stimuli live in a 2-dimensional space, and so this population vector with 2 neurons can have 4 values. So it's kind of a projection. And if you have 3 neurons, you will have 8 patterns, so whatever it was with this third neuron having a value 0, plus another receptive field for the third neuron, another vector, and now if that neuron responds, we will add 4 more patterns. So, in other words, for this vector here represents the preferred stimulus for one neuron. This is neuron number 2, neuron number 3. But if they all live in the same stimulus space, the more neurons you have, the more values, discrete values, the neural population will have. But if you have n neurons, you will have 2 to the n values. But they all have underlying topography based on the structure of their receptive fields, which you can quantify and discretize. Right now I will illustrate this formula in a variety of contexts. But the main conclusion from the first statement is that the claim is that you can modify the population vector by introducing one more factor, and this factor is obtained from sharpness of the nonlinearity. So the bigger the beta, the sharper is the neuron, and such that neural responses that are more reliable should be weighted more strongly, and no other information is necessary. For example, thresholds do not participate in the optimal readout. I showed, I think, this vector on this slide here. This is a different situation where the tuning curves are different. They're still tuned to the same stimulus, and they have slightly different widths. But the differences are caused by changes in the threshold. So they all have different thresholds, but the same beta. And in this case, you can capture what this population provides with the classic population vector. So there are cases where it works, and then there are cases where the population vector fails. And for example, I will skip this, but it also works with, say, noise correlations. But maybe I will ask for questions. Stop and ask for questions. Yes.
The original paper from 2008 showed the 2 curves of the amount of information, and it was based on experimental data, right?
Yes, it was based on experiment. It was, you can think of it as it was analysis of experimental data. There were some asterisks to the experimental data because they kind of realigned the stimulus. So the stimulus, each neuron had a different, each neuron has a different vector W. So they created a synthetic population, imagining how that neuron would respond if the stimulus deviated from its preferred orientation. So there was this, so there's kind of a, because at that time they couldn't record multiple neurons. But there are other papers, for example, from Jonathan Victor lab, where they found the same result with tetrodes from groups of similarly tuned neurons in the visual cortex. And in that case, it's direct analysis of neural responses.
So would you expect the same amount of information from this method and from the words method in experimental data?
So the question is, when do you expect these 2 answers to differ? So if each neuron has a distinguishable vector, then whether or not you scale them by a factor beta doesn't matter, because this is what I will illustrate here. So when does the classic population vector lose information? So you can imagine this case with 3 neurons, and in this case, you need to have the classic population vector will lose information when it has multiple neurons that have the same selectivity but different levels of reliability. So if you have 3 neurons but 2 of them are aligned, but they are kind of different magnitude because they have different noise, then in the information preserving version you keep track that these are different patterns because they have different scaling. But the classical population vector will combine some patterns into one, and which will lead to information loss. Yes? Okay? No? Almost.
So I want to go back to the issue of word versus count.
Yes.
So basically the assumption here is that in terms of time, there is no information because everything here is just probability of spike. But in reality, there may be some information in the time pattern which is absent from the count at all. So the count, I can write down a model where the count doesn't have any information.
Yes. So
and
there
all
is
information resigned in the temporal pattern. Then
Yes.
you need to go to words.
Right. Wait. So there are 2 problems here. One is count versus pattern, and the other one, this is count across neural population, and the other one is count across neural population versus temporal pattern. So let's deal for now with the kind of a spatial or across the neural population, and the temporal. For now we can leave it aside, but one can consider the temporal pattern as kind of a multiple neurons. So, but for the purposes of this talk, and we will leave the temporal discussion, but you can think about how the same equation could apply in time, because I can do stimulus. So right now W was a purely spatial quantity, but W could have time in it. So, but for now, no, count means summing across the population. And we did this analysis using the data that I collected in the visual cortex a while back. So it's a relatively small number of neurons, but we reproduce with real data the same effect, that you can have information about orientation from full population, and also from this information preserving construction, and then the spike count and the classical population vector. I think that goes a little bit directly towards this question, because in real situations you have preferred stimulus patterns that are not exactly the same, but if they are sufficiently close, plus neural noise, then that will lead to some patterns to be not distinguishable, and that's the difference between this curve and that curve. So I will summarize some of parts. We can go into detail, but the claim is that at one moment in time, to read out responses of neural population, the proposal is to modify the population vector by adding another factor that reflects neuronal variability. And actually, so something I glossed over, it actually works in the presence of noise correlations, as long as noise correlations do not depend on the stimulus, do not change with the stimulus. And notice that the population vector has the same dimensionality as the stimulus. So as you increase the number of neurons, the population vector will take increasing number of values, 2 to the n potentially, but it all still lives in the same space as the stimulus. So I think this equation addresses the problems of large neural populations, but does not yet address the question of what to do with high dimensionality of the stimulus. Any questions about this part? No? So now the second part is to what to do with high-dimensional stimuli. So this equation says that population vector has the same dimensionality as the space of neuronal selectivity, and as you add more neurons, you will have higher accuracy. So what does it mean in terms of movement? For example, the movement that the animal is making is three-dimensional. You can have millions of neurons, but the movement is still three-dimensional, and the population vector is still three-dimensional. It can take more values. If you recruit more neurons, you will be able to make more accurate movement, but the population vector and the movement live in the same, say, three-dimensional space. Okay. So now what to do with dimensionality, and so it turns out that technically this computation between vector t, population vector, and vector s, if they are moderately high dimension, this becomes hard to compute quantity from a practical standpoint, because even if this is 10-dimensional, then I have to compute a 10-dimensional times 10-dimensional histogram. It becomes unwieldy. So we found a series of approximations. This is not a claim that they are optimal in some sense, but I will tell you how in practice it works. And so we will start with relatively small dimensional stimuli that live on a sphere, and each dot represents the selectivity of the neuron. And in this direction, we will go from small number of neurons to, say, thousands of neurons. And one can compute full information with the Monte Carlo estimate. That's the black curve. And I will be showing a series of approximations where we deviate from this full information to more practical pieces. So the first piece is, so we need to compute full information between vector t and vector s. Well, one approach is, even though it's vector t and vector s, so we can choose in what direction we want to go, from most accurate to most basic, or vice versa. So I prepared slides from full computation to most basic. So in principle, by the data processing inequality, I'm going to decompose this stimulus vector along with all of its dimensions and compute component by component conditional upon the rest of the other components. So we'll compute information between vector t and stimulus component 1, and then information between vector t and stimulus component 2 conditional upon S1. So there will be a series of these approximations. Then there is still vector t, and we can simplify it and just use one component. So it is an approximation, and this is the red curve here. So it loses information, but not within 80% of the maximum. And then another approximation is ignore these various components and just compute one component versus another component, and we call it a component independent approximation. So there is a full equation and then a series of approximations that neglect more and more of the structure of the stimulus. So the part that is most accurate is this component conditional equation, where you take the power, the component, stimulus component, plus the population vector component, plus the power in the rest of the population vector components. Yes?
When you write 3D uncorrelated stimuli, that means that the stimuli, each stimulus is independent, or the neurons are not overlapping? What is uncorrelated here?
So it means that the stimulus probability distribution factorizes.
So
And the stimulus in this case is conditionally, the neural responses in these simulations are conditionally independent given the stimulus. Otherwise, we cannot compute the full information. But these approximations, they actually can be computed when the neural responses are not conditionally independent.
But the receptive fields of the neurons do overlap here? Or?
So these are the illustration of receptive fields of neurons. So you see that they are kind of symmetrically distributed around in 3 dimensions. So I wanted to show the equation that gives the most, I would say, it's written out in detail, that provides the most accuracy without losing, well, practical. So this is, we call it an isotropic approximation. It's information between stimulus and population vector, disassembled into various components of the stimulus. Each component is computed under the conditional, under conditions of the other stimulus components. And here one can show that this is exact. You keep the same component in the population vector plus the power in other components. So in human language, suppose I'm interested in conveying about the visual signal. So the way I think it's easiest to think about it is think about it in the Fourier space. So S0 will be the mean stimulus, the mean luminance. T0 will be the average neural average response of the neural population. So the first term is information between the mean luminance and the response, the average response of the neural population. That's S0 and T0. Then we think about, well, I add a term, information about the next frequency, which is non-zero, and the same frequency, spatial frequency of neural response across the population, between S1 and T1. Conditional on the mean response, plus information in the next frequency in the stimulus and the same frequency in the population response. Conditional upon the mean and the lower frequency. So you can see where the modulation comes in. The modulation comes in from this conditional probability distribution. So in order to compute this information, I have to compute information between, for example, the first Fourier component in the stimulus and the same Fourier component across the neural population, conditional upon the mean, and then integrated over the mean. So in the context, for example, in the retina, you might be interested in taking information between a high frequency in the both stimulus and across the neural population, conditional upon what is happening on broader scales. So that's the modulation part here. The other part is that that I haven't described yet, is that the population vector, we take the same component, plus it's important to take into account the combined power along all other components. And it turns out that it's sufficient to only take components in the high frequencies. So this has the elements of the complex cell-like computation. And this equation, I think, summarizes the second part of the talk. It says that if I want to compute information between a high-dimensional stimulus and a similarly high-dimensional population response, I have to decompose stimuli into their dimensions, and I also have to compute population vector along the same basis as the stimulus, but also keep track of the power along other dimensions. So I haven't fully derived it, but I hope I can summarize it with this equation. Yes.
So the order doesn't matter?
The order can matter in practice. So empirically we find that this is the slide that talks about the order. So in practice, it seems that the best way to compute is to start with the component that has the largest variance. And this is shown here, information versus in the case of two-dimensional stimuli, and this is the variance in the component 1 versus component 2. So on this side of the graph, the first dimension has larger variance than the second dimension. And in this case, more accurate solution is the solid line. It is when we first compute information with respect to the first component, then the second. So in order that they are largest. And these curves switch at line equals 1. So numerically, we find that you always have to start with the component with the largest variance.
But won't be, if you go back to the slides, it won't be a better way just to, in the normalization toward power spectrum, both for T and S, just to leave out SB rather than SB plus 1, 2.
So you would
no,
like...
SB is fine, but
SB
I'm
is
talking
fine.
about the power. Why shouldn't we just the power of everything aside from B? Then the order doesn't matter why.
So you're asking why is this term is here?
Why not the power spectrum of everything else?
So it turns out so we call it an isotropic approximation, and this isotropic approximation becomes exact in this case where the stimuli are isotropically distributed. And in this case, you can show that you don't need actually components of TP that are less than, like there's nothing B minus 1. So this approximation becomes exact for some probability distribution of stimuli. Now you don't like it?
It should be symmetrized. There should be an optimal way for symmetrizing it. But the order doesn't matter. The expression should be such that the order doesn't matter.
It's possible. I agree. This is not. These are all approximations. There is no claim for it being optimal, but that's the one that we came up with. And I know it's exact in certain cases, so it doesn't. Maybe there's a better way of writing it out. But what I liked about this is that it had some elements of what corresponds with, say, visual processing, with quadratic approximation and the modulation. So now a more practical and maybe easier part to describe is that if we need to compute this conditional distribution of stimuli and neural responses given a broader signal, how to minimize information loss and the specific role of inhibitory neurons. So in the population vector idea, if you have two neurons, and we will be summing, the claim is that you can sum their responses with some weighted factor, and but given that we will be summing them and they will be modulated, it turns out that I don't need to modulate them equally. And so I will be delivering modulation signal to only one of these two neurons. And the way to understand it is that because the sum will be modulated. So if I have two neurons and their activity will be summed, if I modulate only one of them, I can in effect modulate the sum. So I don't need to modulate both in order to modulate the result. And when I don't modulate, I am saving every connection that we make comes with noise. If I don't have a connection, I also do not have noise. So in practice, what happens is that if you want to have a modulation signal, assuming that we will be summing the neural activity, then I'm giving the intuition, we backed it up with numerical simulations, is that the modulation signal has to go to a subset of neurons. You don't need to modulate everybody. And furthermore, it turns out, I'm also giving the conclusion right away, that there is a symmetry. The modulation signal has to go through inhibitory cell. It doesn't, and as opposed to excitatory cell. So if so the way it happens, that suppose you have two neurons that are equivalent up until the modulatory signal was applied. When the inhibitory cell produces the modulation, this cell is suppressed. It will be a low-rate neuron, and this becomes a relatively high-rate neuron. And so when we modulate, the low-rate neuron will be sometimes more suppressed and sometimes less suppressed. And the reason it has to go through the inhibitory neuron is because when you modulate a low-rate cell, it's kind of a form of a stochastic resonance. You create less noise. It's not exactly accurate, but that's the intuition. And I will show the numerical analysis that go with this modulation. So we compare, this is just for two neurons here, information as a function of how much this neuronal pair spikes, the average firing rate. And then there will be two scenarios. One is modulate both of them equally, modulate the cell that has the highest firing rate, and modulate them equally. So what is shown on the left is information as a function of spike probability. On the right is the same graph, but as a percentage of the information under modulation, so a fractional impact of modulation. So it turns out that if you modulate the neuron that is the highest spiking, so modulate with excitation, there will be more loss than modulating them equally, and more loss than modulating the neuron that has the lowest firing rate. So this is modulation equal, this is modulation with inhibitory neuron, and this is modulation with excitatory neuron. Yes?
So before the modulation from the previous part came from the other neurons or other part of the signal, so you need to calculate this modulations.
Yes.
So we assume the calculation itself is unnoisy, so there's no noise in the calculation of the modulation signal?
Of course, there is noise in the calculation of the modulation signal. So this is information in the kind of a primary pathway, in the local pathway, and then plus integrated over the modulational values.
No, but if there's noise in the modulation value, it's probably harder to compute because it's not added, it's multiplicative noise. But wouldn't it still be better to have modulation on more than just one cell, to modulate multiple cells?
It turns out, no. So there are cases, so I will show you, so it's groups of neurons. There are cases where you want to direct to both, and it depends on their firing rate. There's a separate slide. This is with 3 neurons, essentially the same thing, so I will skip that. And I will address the question that Jonathan asked. So under what conditions it is better to modulate both neurons with And then it turns out, so if neurons have identical spike rates, and so if they have a small rate, if they're already in this regime where they're sparsely firing, then it's better to modulate them both. So this is information loss, and modulation with both of them is better. And then when their firing rate increases certain value, then it's better to modulate one of them. So it depends on how sparsely they are located. In other words, so when we apply this to the retina, you form a class of high spiking neurons and a low spiking neurons, and you modulate the low spiking neurons, but it is still a class of neurons. So, and everybody within that class is modulated. So I know it's maybe a lot of material, so I will maybe conclude and then ask for questions and have more of a discussion. So the results that I presented for discussion is that the population vector can be modified in specific ways, which explains why it's mostly correct, but in order to preserve information, one has to take into account how reliable each neuron is. In other words, as Hein pointed out, one has to scale the preferred pattern of the stimulus by its reliability. That's statement number one. And notice that this information preserving readout is still linear in neural responses, even though it's a nonlinear function of the stimulus, but it's a linear function of the neural responses is sufficient to capture information that is contained in the neural population. And for high-dimensional stimuli, there is a series of decompositions that progressively lose more and more information, but the one that we found to be most accurate, even though most computationally difficult, is the one that decomposes the stimulus according to different dimensions and computes a conditional distribution, which is like modulation, and evaluates information between the stimulus component and the corresponding population component while keeping track of the power in other population vector components. So one can think that this is normalization, or sometimes in cortical circuits we talk about local gain control mechanism. So it actually, one could think of how this derives from a general equation for computing information. And a practical prediction is that these modulatory signals have to be delivered to local circuits with inhibitory neurons. And we actually have verified this prediction in the retina, where the role of inhibitory neurons was placed by the amacrine cell, and these were two classes of ganglion cells that had the difference between them in terms of the threshold could be explained by this additional inhibitory input. Thanks for staying till the end, and thank you for your attention.
I have a question about the betas. The beta depends on the tuning function, tuning curve, right?
Yes.
But at least if you think about a single trial, there's also variability around this tuning curve for each neuron, right?
Yeah.
So is that somehow taken into account, or?
Yes. Well, maybe, let's see. Maybe we can use this as a graph. So there are several statements in this curve, in this result. Officially, it is derived as a binary neuron that has two states, 0 and 1, and the probability switches using this function. So the broader this range of where it's switching, so for these values of stimuli, the response is very determined, variability is low. For this range of stimuli, the response is reproducible. Also, so it will be 1 all the time. For this stimuli, it will be 0 all the time. So variability is only in the middle region, because we present the stimulus, and half of the time it spikes, and half of the time it doesn't spike. So this parameter beta, you can think about the range of stimuli where the neural response is uncertain. And so, because this is a specific model, when you specify the probability, you also specify all moments of the distribution, and that's another part. On the other hand, at the same time, you can think of this as a response of the neural population. And so, in this case, you sum the you sum the stimulus, so the parameter beta controls variability. There is another statement that one could have, is that because this is, the claim is that this is the only thing that matters. And the question is why this is not obvious in the tuning curves. Because I can get this parameter from a tuning curve. But in order to get that parameter, I have to plot the tuning curve, take the logarithm of the tuning curve near the peak, take the second derivative near the peak, and do some other transformations and get the beta. So the mathematical expression is not simple when I write down the equation in terms of the tuning curves, even though the information is there.
More question? Let's maybe let's do.
Just an idea taken from your talk, from the previous talk. Do you think that these parameters, the width of the tuning curve or reliability and something like that, do they might be pointing to some hierarchy?
Yes. So the hidden message in this slide is this graph right here. So because the bigger the neural population, the n is 2 to the n. So the bigger is, so what does this nonlinearity does? It compresses the infinite stimulus into the Poincaré circle. And the size of this Poincaré circle is logarithm of the number of neurons. So if I have n neurons, we have 2 to the n patterns. The radius of the map is log of that number. And so I think that the neuronal nonlinearity in just this threshold part is the one that makes representation hyperbolic. So that's the hidden message.
I want to ask about noise correlation. I think that optimal linear readouts will depend on the noise correlation.
So we have an equation.
Is it buried in beta? Beta now has different definitions.
No, it's easier to show you with an equation and to describe when noise correlations matter and when they don't matter.
I'm talking about stimulus-independent noise correlation.
Yes, stimulus independent. So here's a slide about noise correlations. In this curve, in this population, you have a set of neurons tuned to one stimulus and a set of neurons tuned to another stimulus. And they also have different betas, so they have different tuning widths. In addition, we introduced stimulus-independent noise correlations of this form, 5y minus. So they depend on preferred values of these two neurons. In the presence of these noise correlations, if you plot the tuning curves, they look irregular. but we can still write down the probability of the population response given the stimulus in this form, where you have a function of the stimulus, a function of the response, and then e to the minus this vector t, which depends on r, stimulus, plus these correlations. So as long as these correlations do not depend on the stimulus, the form of the population vector readout of information preserving population vector is unchanged. So when the noise correlations go up and down, the information that the neural population encodes goes up and down. But I can read out what it has. But I can read out what there is in the neural population with the same equation.
Any time for one more question? Right, thank you very much.
I agree that the noise correlations
That's right.
Yes, I mean, given that it's so rush, I will be.
תראי לי מה מיני אומר.
No, I can
זה עוד לא מידי אומר?
והרוב.
I think
אתה
it's
בצרפתית גם.
Don't worry about it. If you can just tell me a picture.
Okay.
So that would be perfect. That's it.
Okay, thank you so much.
Your formal problem is now over.
Thank you so much.
Yeah,
So,
Now it's me, no?
what I suggest,
זה אתה?
we
אחר כך.
לא נפגשת ביום
ביקשתי את זה, כיוון ש...
כן.
we have stuff to do.
היא אמרה לי שזה...
אז אני לא יודעת.
מתי התייחס לה?
I can ask Sarah.
Yes.
Is it? Can I visit that museum? Is it still open?
So that's right. You wanted to think about the museum. I think it closes at
Which
very
museum?
time. The Israeli museum.
The Israel Museum?
I think it closes quite early, rather early, but I can check. No problem.
Yeah, maybe I was going to stop by the Israel Museum.
רק שאנשים מעשי ודאי.
I can get the
You can
אין לו זמן כרגע לדבר, בגלל שהוא עסוק עם אורגניזציה שמבין ושצריך לדבר.
לפני שפורמים, לא?
כן, בדיוק, שצריך לדבר והוא מחדד אותך למעשה ורוצה לדבר ואנחנו כולנו הולכים באותו כיוון, שלום, בלה 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 בלה. לא הולכים באותו כיוון.
אני הולך לכיוון של שלום, כי אני גם עושה משהו בנדון.
לא, אבל זה, כן, זה, כמו שהוא אמר לי, קונסטרקטיב דיסקשן, אין קונסטרקטיב דיסקשן, הוא מאמר על החיים שלנו.
expect you
זה מאמר על החיים שלנו, סיבה, מה שאתה מדבר. אז החיים שלנו
the second floor is farthest one from the corridor.
הוא לא מבין שזה, אבל מה בעצם? זה לא עניין של גוד וילדו. זה רציני, זה לא שאנחנו... שאנחנו אומרים,
אנחנו לא גורמים נזק.
לא גורמים נזק, עושים את ההפך.
זה דבר חשוב. לא לגרום נזק.
נראה מה... אני הולכים באותו כיוון, שלום, אני מאוד מכבד את הדעה שלך. פעילות שלי לא טובה. אני יושב שם בפריז, ככה יש לנו דדליין. שתינו זה הבעיה, זה דד, זה לא...
So I will do 15 minutes there and then the corner will be released.
Yeah, okay.
טוב, אז אחר כך אתה מדבר איתי כמה זמן?
אלא אם נלך, אני חושב שהולך.
Okay, as you like.
Or maybe I will stay here if I'm too late.
בסדר, פריז.
Okay, as you like. You can
Because I wanted to talk to people. You understand that's more important than
Excuse me, yes? I'm telling what we did with
I just want to say goodbye and thank you for coming. It was
Thank you
two
very much,
amazing,
Tatyana.
amazing talks. And have a great trip back home.
Thank you.
Sorry that it's so cold. You know, we didn't- From San Diego, you know, it's...
Tanya, what about
בתקופה של הדלת פה,
I'll be in my office from now and
Look at them.
if you come at 5 we'll have a little bit of time to have a conversation.
So you're busy till 5?
I'm busy about 4:15, quarter to 5. Come by then,
אתם נשארים לדבר? It's your stuff.
So, what is the schedule? Can you talk?
You talk with Zianta,
Yeah.
and then we talk. So continue.
I have another meeting at 3:30.
Okay, so go on. You're flying to Paris?
No, I found a direct flight.
Oh, you mean...
But I will be back in Paris in December, or
Yeah, but
we can coordinate. You will not be there?
I'm holding as much as possible Paris.
But when
I
will
was
be-
there I wasn't work for 1 month. So
That's it?
Paris, 2 weeks, and Milano, 10
But when
days.
will you be back in Paris?
I don't know. So you have a flight there late tonight?
Do you want to go to have coffee or to my office?
No, because I'm going to have coffee with her.
So we can make it, we can eat in, we can sit in the room.
So you go now and I will... So at
Or you want to go
3:30, you say. You're not at 3:30?
Yeah. So you want coffee or you want
Or what are the options?
coffee or no coffee?
We can do tea.
Tea.
Well, yeah. So,
Let's go to my office because I want to show you something on the screen. Was it the taxi okay last night?
yeah, he was friendly. He talked to me. He was quite talkative. So it's good that you directed him towards the right path. And,
That's the student was.
the student,
We told you about the student that was murdered.
this
So
is her.
that's yours.
And this is on the outside.
So what did she just? She was just on a.
It was so close,
It
no?
was a chill. I don't know. So what did you do? I don't know. I don't know. So, difficult to say. I think that there are many things that we don't know.
Yeah, I don't know, it's probably even difficult how they're surviving in these towns.
If they survive. If they survive. They're not always in families.
This project focused on developing small molecule inhibitors to PD-1, PD-L1 pathway. PD-1, PD-L1 pathway is an allowed cancer. PD-1, PD-L1 pathway allow cancer to avoid immune system. So, this project focused on developing a small molecule inhibitor to PD-1, PD-L1 pathway. PD-1, PD-L1 pathway is this pathway. This pathway is allow cancer cells to avoid the immune system. Current therapy that also target this pathway are monoclonal antibodies, but they have limitation. Low patient response rate, high costs, and have side effects. This idea is to block this pathway by a small molecule. Well, also this small molecule offer an effective and cheaper solution. This small molecule offer an effective and cheaper solution than monoclonal antibodies. They offer advantages like oral delivery, a simpler production, and higher response rate. Simpler and cheaper production.